DNA separation by gel electrophoresis. First of all, we have to know what is electrophoresis and why the electrophoresis is used. Actually, it is used to separate DNA fragments from each other. It is also used to determine the sizes of DNA fragments with the help of different ladders which are used along with the DNA fragments. It is also used to determine the presence of amount of DNA, that is how much of DNA is present in a specific quantity of DNA samples. We can easily determine with comparing with the different letters and quantified amount of letters along with the DNA samples in the electrophoresis process to analyze the restriction digestion products. That is, by using restriction digestion, we can easily cut the DNA fragments, and these cut fragments can be observed by the process of electrophoresis. So simply, the gel electrophoresis is a method that separate macromolecules, either nucleic acids or proteins. Electrophoresis also describes the migration of charged particles under the influence of an electric field. Again, another thing is that a gel is a colloid in a solid form. Now come to Agarose gel electrophoresis. Agarose gel electrophoresis is a method to separate DNA or RNA molecules by size. This is achieved by moving negatively charged nucleic acid molecules through an agarose matrix with an electric field, that is electrophoresis. Shorter molecules move faster and migrate faster than longer ones. This DNA actually can be visualized in gel by the addition of ethidium bromide only. Principle of electrophoresis. Powerful separation method frequently used to analyze DNA fragments generated by restriction enzymes. So it is a very powerful tool that can easily distinguish, and noticeable differences can be observed while going for this restriction enzyme analysis. Convenient analytical method for determining the size of DNA molecules in the ranges of 500 to 30,000 base pairs. So using this electrophoresis method, we can easily visualize 500 to 30,000 base pair DNA bands. So it's also employed electromotive force to move molecules through a porous gel. So we are using agarose gel, and in this agarose gel, this is porous in nature and employed electromotive force, and these molecules of DNA can be moved through these pores. Separates molecules from each other on the basis of size. So this electrophoresis separate the molecules according to their size and charge and shape. So according to these three conditions, the molecules are separated. Basis of separation depends on how the sample and gel are prepared. So the different graded agarose are used for different purposes of study, and on this way we can easily separate out the DNA samples from in agarose gel.